In this lecture, we are going to discuss the testing and ways to cultivate and isolate the enteric bacteria, or the coliforms, and these are our intestinal bacteria. This lecture has been separated into two parts, and this is part 1. The tests that we're going to discuss are the carbohydrate utilization, ONPG, or orthonitrophenylgalactoside test, indole, methyl red, Voges Proskauer citrate, the triple sugar iron agar, or TSIA, urea phenylalanine deaminase, or PDA, and oxidase. Now, it's very, very important when we're talking about the coliforms, or the enteric bacteria, that you thoroughly understand the tests, because as diagnostic scientists, we are required to understand all of the tests that we're going to be performing. Even if it's a machine that you are putting the organism in that will be performing the tests, you need to understand all of the tests that that machine is doing in order to differentiate one organism from another. And the reason is, the machine may make a mistake, and it's up to you as the scientist to analyze all of the results that come out of a machine and be able to say, oh yes, this makes complete sense. The machine, all the test reactions are correct, and the machine saying it's E. coli, and yes, that makes sense, it is E. coli. You need to be able to identify discrepancies and be able to see a test reaction and say, wait a minute, that test reaction isn't matching with that organism. I need to repeat this test. So it's very important to understand what the tests are, how to perform the tests, and what the reactions mean. So for carbohydrate utilization, this is a test that will test the ability of the bacteria to ferment sugars or carbohydrates. So what you can use are what's called Durham tubes, and actually what you use is a tube that contains media in it that has a sugar. And you can get these tubes with the media with sugar that have Durham tubes. And these Durham tubes will allow you to detect gas production. So the commonly tested carbohydrates are glucose, lactose, maltose, and sucrose. But you can make any type of carbohydrate tube, media tube, and test the ability of the organism to ferment a sugar. Now, these tubes used to be used a long time ago. They're not used in a clinical laboratory anymore. We have our selective and differential media now, and those media will allow you to detect the common carbohydrates that bacteria will ferment. Now in this slide, you have a carbohydrate fermentation test tubes with the Durham tube in it. So number 1 is an uninoculated tube. You see it's kind of got a pinkish orange color with the Durham tube that has media all the way up to the top. In tube number 2, this is an organism that can ferment glucose. So the pH indicator in this media turns from orange to a yellow color. In tube number 3, you have an organism, Proteus vulgaris, that is a gas producer. So it ferments glucose, the media turned yellow, and it produces gas. You see the bubble at the end of the Durham tube. Tube number 4 is a non-fermenter. There's no color change and no gas in the Durham tube. And tube number 5 is E. coli, where you have glucose fermentation and gas production. Now, E. coli and Proteus vulgaris, you expect those to be yellow in a glucose tube because those are Enterobacteriaceae family members, and you guys all know that every single member of the Enterobacteriaceae family ferments glucose. Now, ONPG is another test, and this will allow you to differentiate between late lactose fermenters and non-lactose fermenters. And what this test does is it detects the enzyme beta-galactosidase. And beta-galactosidase will break down lactose into monosaccharides, and it forms orthonitrophenol, which produces a yellow color. So here's an ONPG test. Again, this is a tube test. The broth will be clear in a non-lactose fermenter. It will be yellow in a lactose fermenter. Now, there are some organisms that are late lactose fermenters, and you know that there are selective and differential media that will allow you to identify lactose fermenters. But if it's a late lactose fermenter, it may not turn color within the overnight incubation. So you might miss a late lactose fermenter, which is why the ONPG test is nice, because it allows you to see those late lactose fermenters. Again, this test is not manually performed in the clinical lab any longer, but the instruments that are used in the clinical lab will do the ONPG test. The next set of tests is called IMVIC. And IMVIC stands for, is an acronym for 4 different tests. It's the indole test, the methyl red, the Voges Proskauer, and the citrate test. So for the indole test, this is a test that detects the enzyme tryptophanase, and it's performed in a tryptone broth. So you inoculate your organism in a tryptone broth, you incubate it overnight, 18 to 24 hours at 35 to 37 degrees, and you either add one of 2 reagents, either Kovacs reagent or Ehrlich's reagent. What you will produce, these reagents have the p-dimethylaminobenzoaldehyde, and you will get a red color in a positive reaction. So here you see you have your tryptone broth inoculated with your organism. You add your Ehrlich's or Kovacs reagent. On the right hand side is a negative reaction. You just see a reagent on the top, which is kind of a greenish color. And on the left you have a positive reaction. You get a bright red color when you drop the reagent on the top. Methyl Red is a test that detects strong acid production. You have a glucose broth that contains methyl red. There's a pH indicator so that you get that color change when the media turns acid. So you incubate this test for 48 hours, and methyl red will turn red when that pH gets below 4. So here's your methyl red test, where you have your inoculated broth and you add your reagent, and it will turn bright red if it's positive. Again, this is not a test that's actually performed in the clinical lab, but some of the instrumentations and strip tests will perform this test. Voges Proskauer detects the production of acetylmethylcarbinol or acetoin from glucose fermentation. So since all of our Enterobacteriaceae members ferment glucose, the Voges Proskauer is commonly used, especially, again, not as a manual test that you'll perform in a clinical laboratory, but instrumentation and strip tests will do the Voges Proskauer test. You incubate overnight, you add potassium hydroxide and alpha naphthol, and red is a positive reaction. So here is the Voges Proskauer test. On the left is negative, and the tube on the right in the left-hand picture is positive. In the picture on the right-hand side, the left-hand droppers are positive and the right is negative. The citrate test. This is a test to see if an organism is able to utilize sodium citrate as a sole source of carbon and nitrogen. So if the organism utilizes citrate, it increases or raises the pH. There is a pH indicator, and when the pH becomes alkaline, you get a blue color, and blue would be positive, meaning the organism is able to utilize sodium citrate as a sole carbon source. So in these set of tubes, the tube on the left is uninoculated, so the media it's an agar slant, and the media is green. The tube on the right is positive. This is Enterobacter cloacae, and that turns the slant a blue color. The next tube, Escherichia coli, that's negative for citrate. It didn't turn the slant to blue. And the last tube, Klebsiella pneumoniae, is positive for citrate. TSIA, or triple sugar iron agar. This is one of the most important tests that is manually performed in a clinical laboratory. You're not going to perform this test very often. You might do it if there's a discrepancy or if the instruments were down, but in the past we did perform this test very often, and it is a very good way to differentiate all your different gram-negative bacilli. So what this test does, it tests the ability of your organism to ferment 3 different sugars: glucose, lactose, and sucrose. It also detects the production of hydrogen sulfide, and it also will test the ability of the organism to produce gas. So some organisms, when they ferment carbohydrates, they generate gas. So the sugars or carbohydrates that are tested in triple sugar iron agar, if the organism ferments only glucose, so this will be all of your Enterobacteriaceae family members because they all ferment glucose, the butt of the tube will be acid. It will turn color. If you have either just lactose fermentation or lactose and sucrose fermentation, the slant of the tube will turn a color. It will be acid. Hydrogen sulfide production, the media in the triple sugar iron agar, in addition to having a pH indicator that turns a yellow color when the pH gets into the acid range, it contains iron sulfate, which in an organism that produces hydrogen sulfide, it forms a black precipitate. You read this test after an overnight 18 to 24-hour incubation. So in this test, what you do is you stab the organism right into the center of the agar with a needle, and then you streak the slant of the agar. You incubate it using a very loose tube. Whenever you have tube media in the clinical laboratory, you guys should all remember this from General Micro, you never want to have the caps on the tube screwed on very tightly or the tube will explode, because some of these organisms ferment and they produce gas and will rupture the tube. So you always want to keep your tubes caps very loose, and also you want that oxygen to be able to get into the tube. So in general, for triple sugar iron agar, you have the butt or the bottom of the tube, and then you have the slant or the top of the tube. The butt of the tube is what you read for glucose fermentation. If the butt of the tube does not change color, it means that it didn't ferment glucose. If the butt or the bottom of the tube turns yellow or acid, it does ferment glucose. Now on the slant of the tube, the top portion of the tube, again, if it doesn't change color, it's considered alkaline or K, and it means it didn't ferment lactose or sucrose. If it does change yellow, just the top of the tube, it's an acid reaction, and it means that the organism ferments lactose and or sucrose. So the top, since there's two sugars, you don't really know if it's the lactose or the sucrose, but it's one of those two sugars. So here are actual reactions, and from the left, tube number 1 is uninoculated. So you can see the media is kind of an orangey red color when it's not inoculated at all. When you inoculate it by stabbing it and streaking up the slant, in the next tube, that is considered A over A, or acid over acid. And that big space in the middle means that gas was produced. So how you would read this tube is A over A plus gas. And what this tube means is this organism ferments glucose because the butt or bottom of the tube is yellow. It also ferments lactose and or sucrose because the top of the slant is yellow, and it's a gas producer. So that will allow you to narrow your organisms down, which ones ferment glucose as well as lactose or sucrose and produce gas. In the third tube, urea is another test. So this detects the production of the enzyme urease by bacteria. So bacteria that can produce urease will break down urea into carbon dioxide and ammonia. What happens is when that breakdown occurs and mixes with oxygen, you get ammonium hydroxide, which changes the media to a bright pink color. So here you could see a bunch of tubes. The far left is uninoculated. All the other tubes except for the third tube in are negative. That third tube in, that bright pink colored tube, is positive. Now urease can be a test, or urea media can be a broth test. It can be a slant agar test. It comes in several different forms. But again, you won't do this test manually in the laboratory, but the instrumentation does do a urea test. PDA detects the presence of phenylalanine deaminase enzyme, and that enzyme splits phenylalanine into phenylpyruvic acid. So after an overnight incubation, you add a reagent called ferric chloride, and if the organism can produce phenylalanine deaminase, you get a green color reaction. So here is a phenylalanine deaminase agar slant. You inoculate the organism along the slant. You can see the wave pattern of the organism growing along the slant. After overnight incubation, you'll remove that slant. You can see growth on the slant. You add the ferric chloride reagent, and the slant will turn a nice green color if the organism is PDA positive. Oxidase is actually a test that you will do in the clinical laboratory. It detects the presence of cytochrome oxidase, and most bacteria are oxidase negative. So organisms that are actually oxidase positive are your aerobic organisms. So if you remember, your Enterobacteriaceae family members all ferment glucose, and they are all oxidase negative. So if you had a clinical specimen in the microbiology lab and you knew it was a gram-negative organism and you wanted to see if it was an Enterobacteriaceae member or not, you can quickly do an oxidase test. It's a test that takes about 2 seconds to do, and that's why it's still done in the clinical laboratory, to get an idea of what you might be looking for. So the oxidase test, it's a little dropper, this little oxidase dropper, and you can do this test many different ways. You can pick up your organism using a sterile swab and then drop the oxidase reagent on it, and it'll turn a bright bluish purple, dark purple color if it's positive. Some labs have an oxidase reagent in, and you just swipe your organism onto filter paper, you drop your oxidase reagent on it, and when you get that dark blue purple color, you know it's oxidase positive. So we're going to move on to the second part of our enteric cultivation and testing lecture.